Hi all, welcome to our channel Forensic Pulse Crime to Code. Today we'll be seeing about forensic entomology in determining the time since death. It is one of the most important topics even in MBBS theory exams and also as well as a forensic perspective in determining the time since death in old cadavers. So these are the contents on which we'll be discussing today. So, what is forensic entomology? It is the use of insects and other arthropods that feed on decaying remains to aid legal investigation. This person, he is actually Bernard Greenberg. He is regarded as a father of modern forensic entomology. So, historical significance. So, this is a book called as The Washing Away of Wrongs, written by Sung Soo and translated by Brian E. McKnight. So, here they have described about a case which was solved with the help of forensic entomology. A man was actually murdered in a rice field after being inflicted with multiple injuries. It was suspected that the injuries were caused by a sharp metal object. The investigator of the murder asked the suspects, that is, the rice farmers, to assemble their sickles on the ground. One of the sickles attracted blowflies that we'll be discussing in the future slides. So, probably because of the traces of the blood tissues on the sickle, it attracted more blowflies. The owner of the sickle was interrogated and he confessed to the killing of the man in that report. So here we have seen the case where in previous like 18th century since they have been using forensic entomology to solve cases, and this is another case that was solved in a French courtroom, and this is known as the first application of forensic entomology in the early 1850s. So this case was it was given by Bergred. In that report, it was written that the mummified child was found behind a chimney during the house renovation. The insects found on that mummified corpse were admitted as evidence that current occupants were not the culprits and they were acquitted. So then again, in 1986, Smith categorized the insects into four carrion ecological communities. They are the necrophagus, the predators and the parasites of the necrophagus insects, and omnivorous insects and adventive species. Necrophagus are those that feed on the carrions or the dead bodies. Predators and parasites of the necrophagus insects are those that feed on these necrophagous insects. Omnivorous insects are those that feed on either necrophagus or the predators. Examples include wasps, ants, and some beetles. So, adventive species are such that use the dead body as an extension of their environment. So uses we can broadly classify into urban, store product and medical legal cases. Urban that we use in a household basis like for termite, bed bugs or any some parasite infestation in our household services include urban uses, and store product that is in grain and food contamination by insects, any storage products, that time we use forensic etymology, and medico legal cases for determining the time since death. So, what are the forensically relevant insects? Mainly, we consider the flies, but we also need to know a little about the beetles and the ants. So, about these, it was written in a book called as a Law Faunae des Cadavres that was written by P. Megnin, and he described the insects in this book. Okay, and they called them as des cadaverous. Okay, so flies are the first one to get attracted towards the dead bodies. There are majorly three categories: Calliphoridae, Sarcophagidae, and Muscidae. So how do we remember these? We can remember like sarco is tissue or flesh. Okay, so these are flesh flies. Then we have blowflies and house flies, the common house flies that we have. So in this Calliphoridae, we have two subgroups, the blue bottles and the green bottles. This is a blue bottle. and this is a green bottle. And then we have the beetles. The beetles are found on old cadavers or in dry state. We have the carrion beetles, the rove beetles, and the carpet beetles. And then the ants. The ants consume the small cadavers. The ants attack the body soon after death before the putrefaction begins. A common place for ant attack is around the eyelids, lips, and the knuckles where the moisture is present. No, so eyelids and lips are the places where moisture is present. The lesions are superficial ulcers with scalloped and serpiginous margins. Sometimes they can be mistaken for antemortem aberrations by their position, that is the eyelids and the lips and the margins, and the lack of bleeding or inflammatory changes usually make them recognizable as ant marks. So then we have the rationale of forensic entomology. Why are we studying this? So after death, invasion of an unprotected body by the saprophagous insects and other small fauna come in successional waves. They have a like a life cycle. After this wave, the second wave, like that, they have the waves. Different species of arthropods colonize the corpse at different periods of death. Some species pass through complex life cycle that can be used to determine the least the minimum time since death by studying the stage of life cycle of the insects. The science is inexact and is modified by a number of factors, both climatic and geographical, but in expert hands can sometimes yield useful information about the date of death at a period when the other indicators have ceased to function. These methods have been used since the middle of the 18th century. So, how are we saying that they have been used to the middle of the 18th century? So, last slide we saw about the cases where we have solved, and in the books also they have mentioned the pictures like this. Here they have mentioned the dead body with the insects, and here they have clearly described the places where more insects would be found, like the abdomen or near the eyelid and the ear where the moisture is present. So they have clearly depicted that the insects where the site of the location, and they are known to be like the insects telling the cause of death. They have celebrated the insects like that. So from these depictions we have come to know that these methods have been used since the middle of the 18th century. So when and how forensically in entomology is used for time since death. So when it is used for determining the time since death when it is more than 72 hours or more old. Why it is so? This is because beyond approximately 3 days, that is 72 hours, all the known pathological information will remain invalid to estimate accurately the time of death of the individual. The indicator of this estimate is based on the knowledge that the beginning of the post-mortem interval is considered to coincide with the time when a fly lays eggs on the body, and its end to be the discovery of the body based on the recognition of the life stage of the oldest colonizing species infesting the cadaver. So we'll be seeing about this in the slides. So how we are using the successional waves. It is based on the fact that your human body after death, it supports a rapidly changing ecosystem. So it goes from fresh state to dry state in a matter of weeks or months depending upon the climatic conditions. Using maggot age and development, using this transition, the maggots also will come and develop. An analysis of the oldest stage of the insect on the corpse and the temperature of the region in which the body was discovered leads to a day or range of days in which the insects oviposited or laid eggs on the corpse. This in turn leads to a day or range of days during which death occurred. This method can be used until the first adults begin to emerge. So if the first adult emerges, we cannot find which generation is present, first or second generation. So within the first adult emerges, we can find what time period the body is. Basic assumptions that a forensic entomology makes before calculation of time of death. So most homicides occur at night under cover of darkness where the flies are presumably inactive. Flies will begin oviposting as soon as they discover the body, and faunal succession in and under the corpse will follow a predictable pattern, that is the successional waves they are mentioning. Weather conditions recorded at a site distant from the death scene reflect the conditions at the scene also. Ambient air temperature are the major factors influencing the rate of maggot development. So common sites for sampling, as I have previously discussed in the before slides, we have known that the insects get attracted towards the moisture. So this moisture is present on the natural orifices, eyes, and on the traumatic open wounds at the corpse substrate interface and under the body. I think we can understand the term corpse substrate interface, the place where the body is placed. The body in contact with the soil means that interface, it will attract more insects. In the fold of cloths and pockets, shoes, socks, etc., from the carpet, bag, or material in which the body might have been wrapped, any certain tissue left behind those materials, from the plastic body bag in which the corpse or the remains have been enclosed for transport to the place of autopsy and storage. So from these sites we have to do the sampling. So, evidence collection, practical guidelines, insects can be collected from a corpse or its surrounding in three groups, namely the maggots, the insects from the soil, and other insects. Okay, so the maggots are immersed and killed in very hot, almost boiling water and transferred to a solution of acetic alcohol, that is 3 parts of 70% alcohol and 1 part of glacial acetic acid. So, why we are killing in hot water? We have two reasons for killing the maggots in hot water. This is because the death of the maggot should be instantaneous. So exact moment of death can be known if we are killing by immersing it in hot water. And then another reason is hot water kills the maggots in a relaxed state in which they are fully stretched. So because of this, we can get the measurements of the maggots, and that can be compared with the measurements from the experimental work. If the maggots are immersed in preservative to kill them, this will cause shrinkage and the measurements will be difficult to interpret. So these are the two reasons why we are killing them in boiling water and not in the any preservatives. So then after killing the maggots, some part of the maggots are also reared. Okay, so they can be kept alive on some meat or liver and reared to adult stage. Any puparia, either empty or within the pupae, should be preserved in a specimen tube. All the containers carrying live specimens must be perforated for gaseous exchange so that the living organism get the proper oxygen. Collect specimens from everywhere on the corpse. Soil insects. The soil sample should be collected in specimen bags from which the insects can later be extracted in the laboratory. Each soil sample should be as much as it would fit in the pint bottle. Soil should not be compacted. Storage in ethanol ensures that a later DNA analysis for identification is still possible. This may be necessary if a morphological identification is not possible. So next we'll see about the life cycle of the Musca domestica housefly. So first, the flies lays eggs within the minutes in summers and within hours in winters. So the first instar hatches from the eggs in 8 hours, and to second instar in 1 day, third instar in 1 day, and third instar in 3 days, third instar to pupa in 3 days. So we can remember as 3 to pupa 3 days, 3 to pupa 3 days. So pupa to adult in 5 days. So these are the changes in summers, whereas in winters, as we can see clearly, it will take a longer time for the life cycle to complete. So a maximum of 22 days we can suspect from the houseflies. So the same diagram, it is explained in the life cycle. Summer, it is written as S, and winter as W. So then these are the waves in insects attacking the body exposed to air. So first week, the first wave is the first week. where decomposition starts. And here we can have, I have just mentioned two insects for easy remembrance. The first is Musca domestica and Califora. So the second wave is occurring in the second week to 3 months. Here it depicts the advanced decomposition where fold order starts to begin. And the insects that we see here is Lucilia and Saprophaga. Third wave is a time between 3 to 4 months, and fatty acid formation starts. And here we have the dermestis and the aglosa, the beetles and all we can see. And fourth wave, 4 to 6 months. It is very easy to remember in this order. Second wave, second week, but in third wave and all we'll have the months. Okay. So third wave 3 to 4 months, fourth wave 4 to 6 months, and the fifth wave we have 6 to 8 months. So in fourth wave, 4 to 6 months, the butyric acid fermentation of fat occurs and caseous products are produced. And here we can have Pheophila and necrobia. And fifth way, we have time period of the crops being 6 to 8 months, and ammoniacal fermentation has started to occur, and there is black liquefaction, hydrote and sulfur. And sixth way, we have 8 to 12 months. Desiccation will start, that is drying, and we have Europoda. In seventh wave, it can be from 1 to 3 years. Extreme desiccation is there in the corpse, and the insects that will be found in the corpse will be Tenula. And in the eighth wave, it will be more than greater than 3 years, and there will be only debris left behind the corpse, and the insect that may be there is Tenebrio. So these are the waves that are present in the bodies that are present in the earth, that is in the air, and this is in the earth. So we have only two waves in the earth. Before burial we have Califora, and second wave after burial we have Phylons, Rhizophagus and other insects. So these are my references. So thank you. If you have any doubts or any suggestions, please comment in the comment box. Thank you.